Hello, friends. My name is host Eric. I'm the host of Talking with Famous People, and I'm going to discuss today a perhaps somewhat controversial topic. But the reason I'm going to discuss it is because my point is that controversial topics are where we need to be the most rational. Everybody does, you know. So the topic here is is COVID-19 man-made? The COVID virus man-made? I've been frustrated trying to answer this question because um, I've found a disingenuity across the board on both sides. You know, that people aren't trying to find out the truth necessarily. And that includes a lot of quote unquote scientists. One way or the other, they're arguing a position here. So I've gone and done some internet research here about this subject, and you're going to see stuff on both sides of the issue here. You might say, the fact that you even consider it possible still that it's man-made, Eric, is crazy. Scientists all agree it's not. Well, let's review some of the arguments being made here, okay? So, Bloomberg says the irony is we may never know how and where the virus emerged. China has ignored appeals for an independent investigation. Hammering Australia with trade restrictions after called for one. It's also stalled efforts by the WHO to get top infectious diseases experts into Wuhan. Has prevented the painstaking epidemiological detective work that could provide insight into the chain of events that brought the virus to the bustling capital of Hubei province and how to stop it from happening again. So, Bloomberg says, we don't know. We may never know. Bloomberg says, additionally, in another article, scientists who study the genetics of the virus are convinced it's of natural origin rather than designed in a lab. An accidental release from the research center in Wuhan is possible in theory, but just so implausible, according to Stanley Perlman, who has visited the facility and rates it highly. One reason is the reputation of Xi Zhengli. In 2004, she found a natural reservoir of coronaviruses in a bat cave, and in February, published a paper saying that the genomic sequence of the new pathogen, COVID-19, was 96% identical to that of the one in the cave. Okay. That's, of course, not responsive or relevant at all to whether or not it was made in a lab, but regardless, she told a Scientific American that a review of the genetic characteristics of viruses she's worked with in the lab didn't match those of the coronavirus spreading in humans. In a social media post, the virologist said she would, quote, swear on my life, unquote, the pathogen causing havoc had nothing to do with her lab. And U.S. Secretary of State Michael Pompeo has backed off earlier claims that enormous evidence that the virus escaped from a Wuhan laboratory. This still leaves scientists asking where and how the virus did. So, you say here, one reason is the reputation of this lady. What's another reason? Is that the best reason you had? That this lady said she'd swear on her life that her lab had nothing to do with it? Remember what happened to the whistleblower doctor in China? He died mysteriously. So no real reason to conclude it's of natural origin here at all, provided, but after here they tell you we may never know, here they say definitely natural or more or less definitely natural. So what is this lady whose reputation we're relying on to assume this is the case, and what does it actually look like if you actually go to the article itself and see what she actually said? Meanwhile, she said she frantically went through her own lab records from the past few years to check for any mishandling of experimental materials, especially during disposal. She breathed a sigh of relief when the results came back. None of the sequences matched those of viruses her team had sampled from bat caves. None of the sequences of materials that had been mishandled during disposal? Those are the only ones you check the records for? Ooh, that really took a load off my mind. I had not slept a wink for days. that's that's your reason why it's not man-made? Okay. I mean, so now look here what it says on NPR. Many questions remain about how exactly SARS began to spread to people. Researchers believe it made its way through people directly through the bat or through an intermediate animal. An intermediate animal? Early focus on the source of the outbreak in Wuhan was a food sea market. Regardless, genetic analysis shows the virus began to spread sometime in the fall or winter of 2019, says Robert Gary, a microbiologist at Tulane University. Those same analyses refuted an earlier theory the virus was genetically engineered in a lab. Gary says the reason is simple. The virus infects people in a way that scientists had never seen before. The virus is just really too good at what it's doing, he says. No human being using a computer could do this. This is very clearly a natural process that occurred. In addition, he says there are no signs of human genetic modification in the virus's code. Okay, well, that last sentence is meaningful. The question, of course, is what would a sign of a modification in human genetic code look like exactly? And is it possible or plausible that they could modify a virus such that it doesn't retain signs of having been genetically modified by humans? So let's look a little further into this thing. But this thing here, the virus is too good at what it's doing, and no human could come up with this, is not a good reason for anything, okay? Especially when the exact route remains a mystery, as it says right here. Let's go to this one. Note all these big claims about how it's definitely not man-made. This was published here on the 22nd of December, 2020. The origin of SARS-CoV-2 and its evolutionary relationship is still ambiguous. Several reports attempted to figure out this critical issue by genome-based phylogenetic analysis, yet limited progress was obtained, principally owing to the disability of these methods to reasonably integrate phylogenetic information from all genes of SARS-CoV-2. A supertree method based on multiple trees can produce the overall reasonable phylogenetic tree. However, the supertree method has been barely used for phylogenetic analysis of viruses. Here we apply the matrix representation with... So, okay, the point is... The discovery of evolution and mutation in SARS-CoV-2 was achieved by MRP pseudosequence supertree analysis. But prior to, according to this anyway, prior to December 22nd, when this was published, that people were using a method to determine the information they needed to determine the origins of the disease that was not able, the disability of these methods to reasonably integrate all the information they needed. So, and yet we were making awfully bold claims, weren't we, people of the world? This is not relevant. I looked into it further to see what it was. So, this thing here says, the spike protein of COVID-2 contains 3 hinges that give the globular domain unexpected orientational freedom. And we propose that these hinges allow S to scan the host cell surface, shielded from antibodies by an extensive glycan coat. So, this apparently is the uniqueness of SARS-2 that makes it so contagious. Just FYI, right? Let's go on to the next one. So, now here's Business Insider. This is a nice bullet-pointed outline of this article. One myth in particular just won't go away. This coronavirus is a man-made bioweapon. Note that it's a man-made bioweapon or a man-made virus, or that it either intentionally or accidentally released from a lab in Wuhan, is not a myth. Okay? it's a speculative claim. It's not determined. If I say for sure that it was released, then I'm making an unsubstantiatable claim. But to say that it's a myth that it's man-made, given all the stuff we just read about how we really don't know, is disingenuous. By using the virus's genetic sequence, scientists have been able to debunk this myth once and for all. The answers lie in the spike proteins lining the outside of the virus, its uniqueness, and how poorly this virus performed in computer simulations. In other words, if you wanted to make a virus, you wouldn't have chosen this one because you would have expected it to fail. So they're talking about mapping the sequence, the genomic sequence, and saying the key is we mapped the genomic sequence, okay, and we determined that it's a unique virus that we haven't seen before, right? So Robert Gary again makes his makes his appearance here. We honed in on the parts of the virus we thought were unique. It might play a role in the evolution of the virus, but also the pathogenesis of it. And a couple things stood out pretty quickly when we were starting to compare it with other coronaviruses. Okay. And he says, the narrator explains that it's impossible to manufacture from scratch the backbone of a virus that can also cause disease. You have to work off of an existing virus. And then Gary says, people just don't know enough about what makes a virus pathogenic to be able to assemble that. How would you pick among all the possibilities to get to that last little bit that's going to turn into this worldwide pathogen? What sequences do you think to put in there? I'm about to put in there. The narrator repeats what he says. Simply, there's just not enough knowledge about how to make a new virus that would also cause significant devastation like SARS-CoV-2 has. So creating a new deadly backbone is pretty much impossible. But there's another way the novel coronavirus could have been created in a lab. That would be using an existing virus background or genetic sequence as a starting point. But additions and deletions in a virus leave a trace that can be pointed out pretty quickly, a little bit like removing a red brick from a wall and replacing it with a black brick. You might see an insertion that looks unusual, and you look out in nature and you see there are no other viruses that have genetic insertions like that. We did not see any genetic insertions that are not also identified in nature. So there's no evidence suggesting that it was man-made or laboratory created somehow. Okay, we did not see any genetic insertions that are not also identified in nature. But we do see, right, that this particular SARS virus has an unusual stock domain which contains 3 hinges that give it an unexpected orientational freedom. So we know that it's distinct in some way from any virus before. The question is, how did it get to be that? How did it gain that distinction, right? And Maciej Boni says it probably wasn't inserted in the lab because we don't see any other viruses. We see other viruses that have the same kind of genetic insertion. We did not see any genetic insertions that are not also identified in nature. And yet, this is a unique virus. So it's different than something that exists in nature in some way, right? Now, if they were to mutate an existing virus, then it would have to show significant genetic similarity to the new virus. The best candidate would be RATG13, a bat virus with 96% gene sequence similarity. Gary says, now 96% sounds pretty close, but in genetic terms, that's a pretty long ways away. To put it in perspective, humans and chimpanzees share 99% of the same genome. Okay. So, note how we represent this fact of the 96% thing as we want it to, depending on our position here. Now, this is not an example of that, but this person also says, Alex Burrizo says, the most straightforward explanation for the suspicious quote-unquote genetic traits is natural recombination for other coronaviruses. So in other words, what he's saying is these specific unique genetic traits of coronavirus 2 are better explained by the normal processes by which viruses become novel, rather than some unusual process. Except we can only find a natural virus that's 96% similar, which is miles and miles away, more further away than chimpanzees and humans are from each other. Here's an article. The Begin Sadat Center for Strategic Studies. I don't know what this is here exactly, but this is related to intelligence community rather than the medical science community. One oddity is the uncanny clustering of certain peculiar properties, the common denominator of which is the exceptionally amplified and/or pathogenic affinities of the index virus towards humans. The likelihood that such a clustering took place naturally in the biogeographic region of Wuhan is faint, but the likelihood that it took place inside WIV is high in view of the type of experiments on coronaviruses conducted there for years. This is further supported by a large variety of collateral occurrences, the above cited examples of misconduct, which should neither be ignored nor regarded as coincidental. The complicated interplay between science and intelligence is expected to objectively account for what occurred, but geopolitical forces and geostrategic factors may hinder progress towards clarity. If the only thing at issue was genomic origin or the direct source of the index virus, the whole matter would be much simpler, but the combination of the two supports the likelihood of an unnatural contagion. Close examination of the combination will prove much more meaningful than separate examinations of the two issues. And they show that even assuming intentional release is the least likely, right? Intentional release is basically regarded as unlikely at this point. You've got natural infection, accidental leakage. This is a nice sort of flow chart that shows possible ways, and purple arrows represent ascending likelihoods at different rates which are not indicated, except that intentional release is considered the lowest likelihood, while the green arrows represent roughly equivalent likelihoods. Notably, each of the three variations related to accidental leakage, reading the table vertically, has two alternative sub-variations, either that the virus was the subject of academic study or that it was exported as a potential bioweapon. This is still an open question. A definite precursor virus has not yet been found for any of the mentioned variations and sub-variations, 10 in total. New scientific or intelligence findings may clarify the assessment summarized in the table. Only time will tell. So this seems to be the most intelligent read on the thing that I've read, okay? Let's read up here some of this list of anomalies that are highlighted in this article. The genomic origin of the index virus, the strain that infected patient 0, has been determined to be a Chinese bat virus that underwent extensive pre-adaptation to humans, including continual transmissibility prior to infecting patient 0. The open question is how, where, and when such an exceptional genomic pre-adaptation took place. On the surface, it appears in this instance the probability of human intervention of whatever kind is higher than naturally occurring spontaneous evolutionary adaptation, though it is difficult to comparatively quantify those two probabilities. Another critical issue concerns significant mismatches and errors that occurred at the Wuhan Institute of Virology and collateral Chinese institutions, both Wuhan and elsewhere in China, during the decade preceding the onset of the pandemic as well as thereafter. A question arising from that inquiry is whether those mismatches and errors were cases of negligence or deliberate obfuscation. The latter, unfortunately, appears to be predominant, deliberate obfuscation. The list of anomalies is long and includes scientific papers with incoherent data and findings, unexplained gaps, inconsistencies and contradictions, meaningless and twisted chronologies, illegitimate non-transparency, elimination and distortion of records and databases, obscuring and possible destruction of existing viruses, including the index virus, and genomic material, pressure put on disobedient scientists, physicians, and officials, disappearance of key persons, expedient interchangeability between military, defense, and civilian institutions and other industries. All of this misconduct was allegedly meant, now I don't know alleged by whom, meant to serve one principal purpose: to hamper the tracing of the roots of the index virus. These deliberate obfuscations collectively form a powerful argument in support of the unnatural contagion concept. Definitely need to be considered, right? In further support of this argument, deliberate and systematic obfuscations about the real epidemic impact of the virus during the first months of the outbreak were recently unveiled in detail across 117 pages of leaked documents from the Hubei Provincial Center for Disease Control and Prevention. Wuhan is the capital of Hubei province. These documents are largely marked classified and for internal use only. No reference in the documents to the roots of the virus is made, but curiously, they do mention, quote, facilities at a bacterial and toxic species preservation center. Bacterial could refer to microbial, or toxic could also mean virulent. The debate is very complicated and contains the potential for explosive revelations on many levels, but intelligence communities around the world have stayed surprisingly silent on the matter. The very persistence of intelligence agencies' silence implies that they judge the initial contagion to have been unnatural. Had they concluded that the pandemic resulted from natural contagion, they probably would have made that conclusion public. The U.S. administration indicated that unnatural contagion was likely, but no such statement has been formally released by U.S. intelligence. In fact, I found a statement by U.S. intelligence that releases that, in fact, it is not man-made, that it is natural. So U.S. intelligence agencies have actually chimed in on this point against the side of being man-made. Now, let's go on to the next thing. Here's an NPR article which talks about the following things. 40% of respondents said they believe the coronavirus was made in a lab in China, even though there is no evidence for this. Scientists say the virus was transmitted to humans from another species. 1/3 of Americans believe that voter fraud helped Joe Biden win the 2020 election, despite the fact that courts, election officials, and the Justice Department have found no evidence of widespread fraud that could have changed the outcome. Those are incredibly different issues. Shockingly different. So, first of all, it is incorrect to believe the coronavirus was made in a lab. It's also incorrect to believe the coronavirus, to believe that COVID was naturally occurring. You shouldn't have any belief about it right now because the issue's still open. It's an open question. But I see better argumentation, for sure, on the side of those who are saying that it was made in a lab. And any objective analysis of the argumentation would agree with me. Again, just the argumentation. The most widely agreed upon origin of SARS-CoV-2 based on its genetics is that its ancestors moved around in wild animals, swapping genetic features as they went along before they jumped into humans. Scientists have yet to find the direct parent of SARS-CoV-2 in feral beasts, though its closest relatives exist in bats. So, based on genetics, it's got weird bits from different kinds of animals, so we're assuming it must have moved naturally through those because that's possible, and one typically tries to avoid attributing intent where things can be explained without that, which is fine. But the thing here is, of course, the fact that it's the weakness in the argumentation, right? That's the problem here. The most widely agreed upon origin based on its genetics is this thing, and yet they found no empirical links to that, actually. So when they look to find the virus host in the wild in terms of animals, the best they can come up with is 96%, which, as I've indicated, is miles and miles away. Now, this Dr. Li Ming Yan was a person who hurt this issue substantially. Why? Because she had horrible argumentation. I've looked into her, and she had an agenda. She wanted it to be one way or the other. I don't care which way it is. I want people to think well. The paper goes on to suggest that, this is what Yan's paper says, SARS-CoV-2 contains a unique furin cleavage site in its spike protein. Or murin, or whatever. Oh no, here, furin, yeah. Among murine coronavirus strains, there are viruses with the furin site and strains without. Closely related strains closer than SARS-1 and 2, yet they are all virulent. So this observation says nothing to me about the virus being engineered. A furin site is a short protein sequence that can be recognized and cut by other proteins. Perlman also said furin sites are found in many coronaviruses, and finding it does not surprise us in the field. Okay, so why did it take me so long to find out precisely what this woman had claimed and precisely why that claim is false? Why did it take me so long before I could find media that actually addressed the issue? Shocking. Appalling. So the thing is, was it man-made or not? Was the COVID virus made in a lab or not? My guess is, if I had to make a bet on it, and bet money on it, I would say yes. But that's assuming that there's some methodology by which this bet could be determined, right? I mean, China's done everything in its power to make sure we can't trace the origins of the virus, and that's very suspicious. Now, most importantly, though, I would say the takeaway from all of this is that everybody is getting all sanctimonious about this stuff, right? NPR, shame on you. Shame on you. The notion that that COVID was made in a lab, it has at least as much likelihood to be correct as the notion that it was not. There is no scientist who claims to know where it came from. There are lots of scientists who are making claims that it's naturally occurring and that it came from animals. The only piece of evidence they have to suggest that is that they see bits of, I guess, animal genetics in the virus somewhere. They've hypothesized from a genetic reason that it bounced around between different animal species before coming back to humans. Remember, they can't find the actual source of the virus in nature. So there's not an animal that they pointed to that said this is the animal that a human interaction with an animal caused this to enter the human population. That's what the claim is when it's natural. That I'm some rural Chinese guy and I am going to capture bats because I'm going to eat them, and as I get one bat, it spits in my mouth and gives me this disease. But they haven't found this disease in any animals. The best they can come up with is 96%, which, as I've indicated, is a very, very far distance from this virus. So, do I have reason to believe it's man-made? No. Do I have reason to believe it's naturally occurring? No. I have reason to claim and assert it's an open question. And if I'm fair and reasonable in my analysis, I'm going to say, and it's more likely to be man-made than not. But we probably will never know. That's the truth, and that's how you think well. And you can see, by doing some research on the internet, our primary media outlets are not doing a good job of assessing this issue at all. They insist on knowing what the conclusion is before they do. You withhold judgment on everything until you actually have done the argumentational work. Media outlets.